give it just one more minute because I see some more people trickling in. Thank you guys already for calling in. All right, we can go ahead and get started. I've got a little bit of introduction, so if people call in the next minute or two, it's not a problem. Well, first off, thank you everyone for calling in. We're very excited about today's webinar. And my name is Jessica. I'm one of the founders of the Tropical Restoration Network. Jimena and Adriana are also on the phone online, and we're all very, very grateful that everyone called in today. Today, we are talking about kind of an interesting topic, a little bit off of our normal beaten path of coral restoration. And the topic today will be innovation, unconventional approaches. I think when everyone thinks about coral restoration, we think about coral nurseries, outplanting, some of the more innovative techniques from direct coral implementation. But the managing the ecosystem as a whole is really, really important because we all know that a healthy ecosystem means healthier reefs. So today, the first speaker is Sander van Lopik, who's going to tell us about his work at RoffaReefs from the Rotterdam Zoo. He's working with fish populations, so specifically with herbivorous fish, increasing biodiversity, and of course, this has direct impact on the reef through algae control and improving overall biodiversity. Our second speaker will be Alwin Hylkema, who's calling in from Saba. He worked with Van Hall Larenstein and Wageningen University in research on reef grazers. So he'll tell us about his new projects, about increasing popular reef grazers such as sea urchins, herbivorous fish, and creating space for new coral recruitments. So again, thank you very much for calling in, and I will go ahead and turn the floor over to Sander.
Yes, let me share my screen. So I can't see you right now, but can you see it full screen too?
Not yet. It looks like we're still in PowerPoint.
This is always
Yeah.
otherwise I need to do my whole screen, I think. Okay, let's do this again. Yes, can you see the whole screen now?
Yeah, this looks good.
Okay.
Thanks.
Nice. So I'm gonna tell you something about RoffaReefs, but also our expedition, because basically that is the focus point of this year, and I have some very exciting stuff to share you. First, start with the mission. So, as Jessica already said, we always focus on corals most of the time, but I think there's many fishes that have different roles, and to explain that a little bit, in Rotterdam, where I come from, also called Roffa in slang, I always use the metaphor of the city. So basically, in the city, you also need people to make sure that, for example, the toilet doesn't break down or surfaces. And these ecosystem services in this manner can be brought by different species. For example, also groupers. I always call them the rodent killers of the coral reef. But you also have the grazers that you can call the gardeners of the reef. And I think most of the time we tend to forget that we should not take them for granted. And that was the main focus of RoffaReefs. How can we make sure that we at least know something about how they work, and also see if we can maybe make more of them. Yeah, like I said, most grazing fish are crucial for the health of ecosystems. They can really prevent phase shifts, and the same is also for sea urchins, but that is for Alwin, of course. And I think sometimes we take them for granted, and I really wanted to learn more from them and also see if we can make more of these fish. And in order to do so, we work with a three-pillar approach. We have a nature-based solution, which is our breeding system. And I also call it the UFO sometimes, the unidentified floating object. And what it does is basically we fill it up with eggs and then see if we can increase the survival rate of these fish eggs. But if you want to know what you're putting into the system, you need to know which egg corresponds to which fish. That's our science base. So I will tell you a little bit more about that later. And of course, if you want to put something back into nature, you also have to see if it doesn't get out, caught out on the other side. And that was the first aim of our local base pillar. Just talk with fishermen and see what they know. And we got next to a unique friendship, also a very cool way of working there. And this is basically how I think nature conservation should always work. But most of the time we focus on the first two. And it is very hard sometimes if you implement the stuff that you do to also really make it local based. And I'm really proud that we're doing that right now. Because basically these fishermen are the stewards of the sea. And in Dutch, we have a very fun saying, especially in Rotterdam, we say this, it's called niet lullen maar poetsen. So the first thing we did is basically see which people are actually working with their hands with fish. So niet lullen maar poetsen means quit talking, do something. And the fun thing there is that if you look in Bonaire, for that matter, there's only two organizations that you need to talk to. That is Piscabon, the Fisheries Union, and Stinapa, the National Parks Authority. And the first thing we did is just do workshops with them and see if we can make a common ground. And that was quite easy because everybody wants a sea full of fish, maybe from a different perspective, but in the end, we formulated a goal. And the cool thing after that is that fishermen have a lot of knowledge, but the fact that it's never been written down doesn't mean it has no value. And one of the things that we did after that is do a workshop with them where we took the calendar of the year, as you can see on the fourth square, where we basically asked them, okay, which fish do you catch and when do they have eggs in the belly? And it is very simple, but it was also quite effective. But the fact that we could do this is basically because I'm not a researcher. I always say I'm an inventor. And the data was not used to put out into the world, but for our own management and also for Piscabon. And that was crucial to do this workshop because in this manner we can show them that it's their knowledge. We were just the lucky ones writing it down. But it was really fundamental for the research that we do on the fish eggs. Note the title, by the way, for anybody that saw Finding Nemo or Finding Dory, you know where it's coming from. And one of the fun things there is if you're working with the fishermen, sometimes they also give you the perspective of the island. And I think that is really crucial. That's why I think it's good that Martin, the chairman of Piscabon, can also tell what he saw when I was coming to the island for the first time.
Er komt iemand van buitenaf, Nederland, waar dan ook, die gaat mij vertellen, eilandbewoner, ik ben hier geboren, getogen, familie, alles zit hier. Die komt van buitenaf mij vertellen hoe ik mijn eiland moet behouden, beschermen, alles aan doen om het zo lang mogelijk open te houden, goed te houden voor mijn kinderen, kleinkinderen, et cetera. Dat is gek voor woorden eigenlijk. Dat ik het zelf niet eens zie. Het is nog gekker voor woorden als je kijkt bestuurlijk gezien. Dat degene die dan in het bestuur zitten van het eiland, dat niet beseffen. Dat niet tegen jou zeggen van, hé, ik vind het tof wat je doet. Wij pakken het hier op. We gaan er wat mee doen. We gaan het pushen.
Oh. And my conclusion from that is always that if you can't always work with the government, but bypass some way, somehow, your way of working to also show people that what you do actually already makes a difference. It's not that you don't need them, but you can put the 3 pillars back together, but also include people that normally otherwise would never work with or for you. And one of the most important things that we did there is during our expedition, we also included a fisherman. So basically the fishermen of Bonaire were the first ones that actually saw the fish eggs that we are catching. And the coolest thing there is that it started all in the Rotterdam Zoo. So I started as an aquarist. And in an aquarist, you always, as an aquarist, you're basically a zookeeper underwater. And the fun thing there is that you learn how to work within these smaller ecosystems, and especially in aquarium, which sounds really weird for a kind of zookeeper, you learn how to think out of that box and do ecosystems. Because if you're managing coral reefs, you see the roles of the fish, and you also get a bit of integrated product design study next to it, because you have to be really resourceful. And that resources we use to do our expedition. So what you see there on the left is our own home-built planktonic nets with some flasks behind it. So you can catch these fish eggs either active or passive. And if we do this actively, we, for example, go to a spawning aggregation and scoop the eggs right out. Then you don't need a DNA analysis, but you really need a good photo ID. And in the end, we want to put it all together to make the first artificial intelligence to recognize fish eggs. And we're already quite far along the way. And the setup is actually quite easy to work with. It's basically a school microscope that we found quite good to work with and is not that expensive. And the only other things you need are just some tools to pipette everything and a screen. And this picture was taken the first time we were in Bonaire, and it was actually in our living room because we didn't have a lab back then. And the fun thing about it that for 2,000 euros, we managed to make a pop-up lab in Bonaire. Together with the WWF Innovation Fund, we could actually do this 4 years ago, and now we have our own lab. But it also shows how easy it actually is to work with. And the cool thing about it is the blue thing that you see on top is actually a camera and a computer in one. So on the screen, you can see a small scale bar in the corner at the bottom. And every picture that we take has this stamp. And that's crucial for example, the artificial intelligence that we have, because now we have an automatic measurement tool that we can hopefully put online for you guys even to work with on our website, expedition.org. And to give you a small tour, this is our website. We just launched it 2 weeks ago, so maybe there are some flaws. If you see any, just let us know. And the first thing you see is the first 24 hours of the life of the yellowtail parrotfish. And as you can see, it comes from cell division till coming out. And I think one thing that we sometimes forget is that we can measure new life and thus hope for coral reefs, especially for fish. For over 40 species, we identified them as an egg for the first time, which is actually amazing that we don't know that much about it. So whether you're a diver, a fisherman, somebody that works in an aquarium, or a scientist, you can contribute. I have to say, aquarist and scientist profiles are not entirely ready yet, but for all the divers in the group, you can make an account, I can give you authorization, and you can already work with it. And the cool thing about it, it's basically a citizen science platform on reproduction of marine fish, and it was not there yet. And if you would say that 5 years ago, I would love you in the face, but I'm really, really glad that we have this right now. You can also log in with your Microsoft or Google account, and it's very easy to work with. So basically, if you saw a fish spawning, you can log in the dive site. By clicking on the site, we already have the longitude and the latitude, so we know where you did your observation. By putting in the date and time, we know what the weather was at that time, and we can do something very cool with that, but I'll show you that afterwards. If you put in your dive depth, Jeroen Ham in Bonaire, a scientist, can actually also go there and see if we can catch some fish eggs. You can fill in the species, the number of the group spawning, if it was a group spawning observed, and you can put in the egg that you saw. But what we are looking for right now is basically the spawning in the profile. So this is quite a flaw in the video. You get some funny image of a cat, and we thank you very much. And backwards at the other side of the database, we can ensure that we can catch fish eggs at this same place. And it would be very cool if we can find other scientists that also work like this. You can also look backwards at the identifications that you did, which is also a great tool to work with. There's some more information on it, but that is not too much new stuff for you, I guess, but it's good for divers in general to actually know what we're doing. There's also a small video of how it started in the zoo if you want to know more about it. And for every profile, we really have partners in Bonaire to thank for. So, for example, for the divers, we do a lot of that together with Dive Friends Bonaire. Piscabon for the fisheries module. For the aquariums, we thank the European Association of Zoos and Aquaria and Burger Zoo in particular, because even there we identified fish eggs for the first time. And for the scientist profile, we're working on it to do that together with the University of Wageningen. And now the question comes, what happens if we don't catch this fish yet? And it sounds like a very weird question, but I think this is really where what we are doing is going even beyond the scope of the fish eggs themselves. Because more than half of all ocean life starts as a floating tiny egg. It is for corals, it is for crabs, it is for lobsters. And one of the cool things that we do with these weather modules is, for the people that went to Bonaire, this is dive site white slave. You can see the saltpeter over here, and this is burnt vierkant, is also predict where these eggs are going to. So an egg is in this manner, a Lagrangian particle, which means it has no motion on the surroundings, but it floats. And most people think that they float very far away, but as you can see here, as the hours go by, these squares that you see on the screen are 2 by 2. So they only go 6 to 8 kilometers far till they hatch, which is actually not that far. And I think if we have models like this, you can do a lot for reef conservation, even tracking forward if you outplan the coral where it would end up if you're looking at the F1 or the F2, which is really something that we need to do more. Not work in nature conservation projects, but in programs. And that's what we try to do here. Because basically we're measuring new life and thus hope. This is the egg of a parrotfish. And I think it is already very cool to watch, but what you see here is a parrotfish getting out of the egg. And the cool thing there is these hatching videos, actually every single time we see them, really makes me the same kid as looking here at frog spawn, for example. And it's amazing that you can actually do this for the first time with only $200 and some nets, and you're ready to go. And I think that also shows that sometimes it is also about making sure that you have the effort, and then afterwards you can make science. And I'm really urging you guys to also work the same manner. Even if you only do it once a year, you can contribute to what I think is one of the coolest things, but I made it myself. So I'm a bit biased there, of course. Does anybody have questions?
Thank you, Sander. I think I speak for everyone when I say that was really interesting. I'll get it started with the first question, and then if anyone else has a question, feel free to put it in the chat, or you go ahead and unmute yourself and we can ask. But Sander, when you collect the eggs, most of the group that we have here today, a lot of them work in mangroves and seagrass. Have you been able to collect eggs from mangrove or seagrass areas?
No, so that is one of one of the fun things is multiple times we say it's a nursery mangroves or seagrass. But there's only one scientific paper that I could find for pelagic spawners where it is actually the case that they were spawning over there. And that is also why I'm really curious if somebody can enlighten me on that. Because if you see a juvenile fish, when it starts as just a half a millimeter big, then my question is it a nursery or is it kindergarten or maybe even the bike rack behind high school? We literally don't know. And this first phases of fish, which is basically the livelihood for a lot of people all across the globe, are really unknown, which is really weird. So we tried it in Bonaire, I think 8 times, and we didn't catch that much. So we focused back onto the reef. But if somebody can do that with more riggedness, that will be really nice for us, because there's a lot to uncover there. Which also doesn't mean, by the way, that we think mangroves or seagrass are not important for fish. They just have a different role, and we need to change our perspective on these ecosystems. We just don't know enough about them.
Yeah, that's super interesting because we do say it all the time. I know I'm guilty of it. We always say it's the nursery area, but it would be nice to actually to prove that. So that's interesting. Is there any other questions out there?
Yeah, I have a question. Can you hear me?
Yes.
Yeah.
Yeah, I just typed it in the chat, but I can also say it. Thank you for your presentation, Sander. I really like it, and also the platform you made. I wonder, with this platform, so you generate a lot of data and new insights with how these eggs distribute. Do you also already have a plan how you will loop back this new information to the fishers and to make it relevant for them? How do you see this?
Well, that depends also on the place. So for Bonaire, I know I can do this actively, and I'm there. But for other places all around the globe, we're basically looking for people that also first want to make a science profile, and if they have that, ask them if they want to be admin, and see if they can make tailor-made work there. So for Bonaire, I know that, for example, Martin, that you saw in the video, is even pushing for more inclusive fisheries management that goes with seasons, as we already do with lobsters. But a good example is that, for example, for blackfin tuna, we catch the first egg in the Caribbean, and there's only data for West Africa. So there's even scientists saying that they only spawn in West Africa, and we just proved that that is not even the case. So even there, I think it's all about, if you connect early enough in doing this, it is basically you're going to school together. That's what we did with Piscobon. So for everybody that wants to do the same, I urge you to actually invite fishermen and let them see the fish eggs first, because then they're already amazed by the fact that they also don't know how it works. And if you also stated in the beginning, then there's a different way of collaboration that I could never dream of. For me, it was logic. Working in a zoo, you always do that because you work a lot in practice. But I think that is really vital if you want to do similar work.
Okay, thank you. Yeah, I agree. I was just thinking maybe they become more wary as soon as these results may yield conservation issues, that there will be marine protected areas in which they're not allowed to fish anymore, and then they may feel like they've been ingeluist. What's that in English? Set up.
Yeah.
Yeah.
No, I get that, but I think then if you include them early enough, they get what you are trying to say, and I think that already helps. It could even be that we already know that a marine protected area works, but if there's seasons in your fishing, and my hope is still that together with Piscobon, we already pitched this to the OLBA in Bonaire, the government, is that if you know that a fish is spawning that is vital for our ecosystems, but also for livelihoods, make sure that they catch fish eggs together with you instead of fish. And if you can do that together, then you can even make a fisherman a conservationist. And maybe I'm too optimistic sometimes, but I still believe that it is possible if you work in the correct way.
Yeah, cool. Your enthusiasm is contagious, so keep it going.
Thanks.
All right, I think we have time for maybe one more question. Is there another question out there?
In the
Yeah,
chat, I see
one
one.
from Alessandra. Hi, Sander. Working in the Caribbean in a very different environment where there is no past record of data such as fish aggregation site and fishermen often not sharing information. Where would you start from if we were to start collecting that data? Well, I think first already go to divers and also fishermen. I think if you start making them feel like conscious of the fact that they know stuff that you don't know, that really helps. But I also have to admit it helped that I came from the outside. So basically, one of the best choices that I made tracing backwards is not living on Bonaire. So I go back and forth, and I can give a more objective view on things in their perspective, which is not necessarily true, but it helps. So it could also be that somebody that is not native to your country comes in, does this stuff and does the workshops. I think literally, and I think Jessica can maybe approve on that. That really helped in the case of putting RoffaReefs down there. I could show them that I had an idea that I want to do it for the island, but not for myself in terms of being in competition for jobs or anything. I don't know if that helps, but the fact that you already have a platform where you can show people that their knowledge has value, that is already a very different start from the conversation. But of course, I only did this now in 2 places and mainly on Bonaire. So of course there will be differences per region or island.
All right, and then I'm actually going to squeeze one more question in because
Okay.
Eden had a question. Do you want to ask it yourself?
Okay, good morning. Thanks for the presentation. I just wanted to find out, like, how do you actually identify the egg to the actual species? Are you guys using or working with the university to identify it using molecular techniques or through other mechanism?
Yeah, we do
And
this in
how
2 ways. Okay, sorry.
uh,
Yeah, and
the second part of the question is, how do you keep these eggs alive in a lab? I mean, will you be able to do it with a basic laboratory settings, how difficult it might be for others to
yeah.
replicate your work and contribute to some of this work?
Well, so we do a lot of work together with I forgot the correct name of the organization, but they work on the heat tolerance. So we also do even stressor test on there. So let's start with how easy it is to keep them alive till the hatching. That is quite easy. And I think there's a reason for that because as larvae, they're really fragile. And the first stage, they're just in their shell. So there's not much that can go wrong. The only thing that you need to make sure of is that there's no predation. So, for example, normally we know most data from fish that we actually eat. So Atlantic herring, for example, in 24 hours, around 2% survives, which is not that much, but that's on average what happens to most marine teleost pelagic spawners. And the fun thing there is that our survival rate is most of the time 50% and up after 24 hours. That's also where I think we can make a conservation difference if you are aiming at the right species, of course. So it is actually quite easy to hatch them, and you can do that in just small containers in the lab. Because when they hatch, that's where the problem comes in. Then there's contamination coming because they're hatching, and especially after the phase where they observe the whole egg yolk, then it can be really tricky. But the first part is actually quite easy, and you will always have some survival, especially if you see a spawning site. And that is basically tracing backwards to the techniques, what we do right now. So we do it either passive, then we always need a DNA analysis, or if we already catch them enough, we can use the artificial intelligence tool. So for some species, we have more than 1,000 pictures, and then it's relatively easy to at least say at family level, for example, that we have a certain fish. For others, it is more difficult, and that's why we need to use DNA if we randomly collect eggs. But I think the coolest thing you can do here is, even if you don't have the DNA sampling technique, if you see a fish spawning aggregation, you know it's there. You just go there with your net, swim right through. I would say maybe it's even good for your skin, but I'm still bald, so I'm not sure about that. And the fun thing there is that you already know where it's coming from. So you just grow it out in the lab overnight, take the picture, and we already can do a lot of morphological work on your egg. And it would be really nice if people all over the world would do that. Not only in the Caribbean, by the way, because this is literally something that you can do even here in the Netherlands, in the North Sea, or in the aquarium behind me, or in the Pacific.
Awesome. Well, thank you so much, Sander. If anyone wants to get a hold of him, you can Google RoffaReefs. If you forget the term RoffaReefs, feel free to reach out to us on social media or through email, and we'll make sure you get all the right contact information. All right, so now we're going to move down the water column a little bit. We're going to switch over to Alwin, who's going to tell us a little bit more about reef grazers.
Yeah, thank you, Jessica. And yeah, happy to be here. Thanks for the invite. I'm happy to tell you more about our work. My name is Alwin. I coordinate the Reef Grazer Research Group on SEBA. I work for Van Hall Larenstein and Wageningen University. And yeah, our main aim is actually trying to influence what you see on the picture, the permanent tug of war that's going on the reef, in which algae compete with corals. And we try to help the corals a little bit by introducing or enhancing native grazers that graze away the algae. So I'm going to tell you a little bit more in this presentation about why I think this is needed, especially in the Caribbean, and or attends the different species that we work on and or future plans. So why do we need more grazers? This is a plot, coral cover on the y-axis and time 1970s till now on the x-axis. And I'm going to plot two hypothetical reefs in here, one from the Indo-Pacific, in which you see large variations in coral cover. It goes up and down. After disturbance, there's often a period of recovery. So this is often seen on Indo-Pacific reefs, even with all the threats that face coral reefs at the moment. And in the Caribbean, we see much more often a pattern like this, a pattern of stepwise degradation. So there's a disturbance, coral bleaching, coral disease, coral cover goes down, and is then stable for a period of time till the next disturbance hits. So why are these two reefs so different? First, let me show you that I'm not entirely making this up. These are just some peer-reviewed studies. This is Indonesia. They reviewed all different studies that were performed across the archipel. And you see that coral cover is indeed fluctuating hugely. And this is the Central Great Barrier Reef in Australia. And you see the same thing, it's going up and down. I'm not saying, let me be very clear about that these reefs are not in trouble. These reefs are also facing a lot of threats, and coral cover is not the only metric that determines the health of a reef. So of course, species composition might change, genetic diversity might change, and productivity could change. So these reefs might not be the same as 30 years ago, but at least there is recovery happening after disturbance. And that's what we often don't see in the Caribbean. So why is that? Why don't we see that? Well, one of the most important reasons that we see very little recovery, Bonaire has been one of the exemptions the last few decades actually, is macroalgae. Macroalgae are very intense competitors of corals. They can overgrow and kill adult corals, especially if these corals are weakened by disease or bleaching. And more importantly, they prevent coral recovery. As you can see on the picture on the right, that coral actually managed to settle, but is now already overgrown by thick mat of Dictyoda. And often the Dictyoda prevents coral recovery or coral settlement altogether. So if we compare the Caribbean versus the Indo-Pacific, we see that the Caribbean have a lower overall herbivore biomass to start with. There's simply less herbivores. And at the same time, we have actually higher algae growth rates because we have more nutrients and we have more trace elements that can also limit algae growth. Also, there's less functional redundancy in general. And what that means is that there's not many different species that occupy the same niche. So, for example, in the Caribbean, we have two Acropora species. Well, certain places in the Pacific have over 100 Acropora species. And in the Caribbean, Acropora is the only large branching species. Well, in the Indo-Pacific, there's a couple other genera that also fulfill that role. So there's much more functional redundancy, and the same is true for grazers. So certain grazer groups that are present in the Pacific, we simply don't have them. So we are dependent on a smaller group of herbivores. And that became, of course, very obvious when the Diadema sea urchins died off in the 1980s. I'm going to tell a little bit more about that later. So all these things together result in a lower resilience. And resilience is the ability to cope with and recover from disturbances. So we have a lower resilience. There's less ability to recover after a coral disease or bleaching event. And the problem of all of this is also that it sort of reinforces itself. So because coral does not recover, there's more substrate available for algae, meaning even more algae, lower grazing pressure, and so on and so on. So what can we do to increase that resilience? Well, first of all, on many islands, we could increase the water quality, prevent runoff, erosion, sewage entering the water, etc. We have to be very careful with our herbivorous fish, prevent bycatch, and we could also enhance the native invertebrate grazers. So my group, the Reef Grazer Research Group, from the SEBA Research Center on SEBA, focuses on approaches to enhance these native invertebrate herbivores. And the species we started with will probably not be a surprise to you. That's the Diadema sea urchin. So Diadema antellarum was the most abundant herbivore till the early 1980s. And then a mass mortality hit in the entire Caribbean. And as a result, 98% of the individuals succumbed because of this disease. So this resulted in an increase in algae and a decrease in CCA and coral cover, and also in reduced coral recruitment. So reducing the resilience of these coral reefs. Recovery of this species was very slow to non-existent, but in the places where they did recover, we saw a phase shift reversal, meaning that these trends in algae, CCA, and coral reversed. So actually coral came back, CCA came back, and coral recruits had a higher survival and a higher growth rate. So those changes were very positive. And that made us and many other people think, okay, we have to make it possible to restore this species. However, they were known to be unculturable. Well, that is not true anymore. Both us and the University of Florida developed culture methods in 2020. So we can now consistently culture this species. And we do that on a shaker table, which continuously keeps the algae or keeps the larvae in motion, similar to what the ocean does. We feed them, and after up till around a month, they are competent and they can settle. Most of them take longer than that, more 40 to 50 days. So in the pictures here at the bottom of the slide, you see the different morphologies depending on age. And especially in the middle, you see day 19, you see a larvae with very long arms. This is still giving us challenges because this makes them much more difficult to culture than other sea urchin species. These arms are very brittle. They break off easily in the process of cultivation. And then the larvae or the arms can regrow, but often the arm gets infected and the larvae dies. So it results in lower survival. So one of our current focuses is upscaling the diadema culture. We now work with conical tanks, that you see in the picture, with an airlift method, and we can culture up to 100,000 larvae per tank. But we still have remaining challenges. We cannot culture all 100,000 larvae to competency. So larval survival is still something we want to improve. Then there's the settlement stage, and it's a normal process, very critical in the life history of the species. So it's normal to lose a lot of individuals there, but still we would like to get those numbers a little higher. And then restocking has been a challenge for us as well, because lab urchins, especially the urchins we started to put out 4 or 5 years ago, were not adapted to live in the wild. And at the moment we work on improving that, and most improvements are relatively simple and we can even do during the grow out in the lab, such as higher water quality and more water movement makes them much more resistant to predation. So a short video of the latest restocking that we did. So yeah, so you can get an idea of the process of restocking these urchins. But of course, in 2022, there was another diadema die-off, also Caribbean white, also very high mortalities in certain places, and a strong reduction in natural settlement rates, which reduced the ability to recover naturally. The responsible micropredator, it's very small unicellular animal, is a scuticociliate. And we are not entirely certain if it was in the Caribbean before the die-off. It was probably not, but it's very hard to prove that. However, we do know that it stayed around after this die-off. It's also staying on benthic surfaces, coral, algae, etc. And we have seen flare-ups of the die-offs after 22. So we think that diadema will remain a vulnerable species, although it's naturally the most logical species to restore because it has been abundant in such high numbers. It might not be the most practical to work with. And more importantly, it's not a good idea to only work with diadema. So that's maybe the most important message. It's important to have a diverse grazer assemblage, because then you're not so dependent on a single species. So we started a reef grazing project 2 years ago to explore the suitability of 3 other species that we thought were suitable for reef restoration. The Caribbean king crab, the West Indian sea egg, also called zeeappel or white sea urchin, and the whelk, West Indian topshell, or kiwa. So here you see on the picture, you see an experiment where we did some work with Trypnostis sea urchins. We put them in a standardized artificial reef unit, and we look at their effect on the benthic community and on corals. And we also did some retention experiments. These are whelks that were also put on artificial reef units. So I'm going to show you some of the results for every of these species. So for the crab, it was already proven by researchers in Florida that they are fantastic herbivores and they can really reduce the algae cover and increase coral growth. But what wasn't known yet is their interactions with other marine organisms, especially with other grazers. And that is important if you want to restore this species together with sea urchins, for example. So what we did trials with more than 50 individual crabs, and we put them in large basins together with other herbivores. And we videoed that for a full night and looked at interactions between the species. So this is a short summary of what we saw. So we saw that quite a few crabs were actually interested in the other grazers. It wasn't easy for them, but sometimes they managed to turn around trypnosis sea urchins, for example, here, and then they can access it to the oral site, and they just empty it. There was not many of these lethal interactions, but what we saw a lot, especially with the diadema, that they also clipped the spines of the sea urchins, and yeah, that's also not so great, because if they do that in nature, it will probably not be lethal. The urchin will just move away, but it means it also moves away from your restoration plot, and that's often not what you want. You want it to stay more or less in the area where you put it. So we think perhaps could still be used for reef restoration purposes, although some further research is needed to look at effects on other organisms, because that's something we didn't study. But they shouldn't be restored in places where there's still a decent sea urchin population, because they will probably have a negative effect on them. And it also doesn't make sense to enhance them at the same moment on the same reef as other grazers. So then the sea eggs, the white sea urchins, Trypnosis ventricosus, they turned out to be really good grazers. You can see in the photo what they did to this reef, standardized artificial reefs that we use. And they also has a positive effect on the coral survival. We would put small coral fragments on top of the reef, and if you look careful, you can see them. And the grazing had a positive effect on them. A similar effect as diadema sea urchins. We compared it with the diadema. So that's very positive. The culture is, well, I wouldn't say easy, but it's definitely easy compared to the diadema. They don't have these long brittle arms, which is great. And their larval culture is also shorter. It's 4 to 5 weeks, while diadema can be double that amount of time. So that makes it really a lot easier. So we started culturing them end of last year, and we now have the first settlers. You see one of them in the photo in our grow out tanks. So we hope to do some co-culture experiments later this year, in which we want to see if the small sea urchins have a positive effect on newly settled coral recruits or transplanted microfragments. So we're positive about the trypnosis. And then we have the whelks, the Citarium pica, which also did pretty good. They removed turf and macroalgae, but we also found out that they eat different algae and bacteria than the other grazers. They also eat cyanobacteria, we found out, which is great, because that's something that nobody likes. But these snails, they eat them. So we did different experiments with them. On the left, you see a small cage on the reef so we could measure their effects on the reef. And on the right, we see a retention experiment where we release them, and you see that there's quite some CCA that is now visible because the snails grazed away the turf algae and the macroalgae. So also positive results here. So what's next? At the moment, we're working in the Triple R project. Triple R is triple restoration approach, and we want to determine if combining coral restoration, grazer enhancement, and placement of structure increases the chances of success of these restoration attempts. So in the picture, you see a very simplified version of the ID. We want to make a more complex structure, also a larger structure. but with sea urchins and with corals on them. So that is our current focus. And then we're working on setting up the Reef Grazers 2 project, which will be a follow-up on the Reef Grazers 1, in which we want to continue focusing on the sea eggs and the whelks, because we think the crabs are not super suitable for our islands. And then we want to focus on the production. They can be cultured. We already cultured them, but the culture can absolutely be optimized. There's a lot of questions about retention, like what is the ideal size, what depth, habitat do they do well, in which habitats do they not do well. So a lot of questions there. And we want to look into the diet. Like I said, the snails, the whelks, they eat different things, but we don't know exactly how it's different, and how they can be complementary to each other and to diadema in restocking, and to create like a diverse grazer assemblage. So with that, I would like to thank all our partners, all colleagues that helped, and the students and volunteers that help us with our work. Thank you so much. We could not do this work without you. And as a last page, before I will take any questions that you might have, I will also share a QR code to our LinkedIn page. So if you're interested in this work, you can follow us there, and we try to post regular updates.
Perfect. Thank you so much. What an interesting presentation. I think algae is something we've all battled with no matter where you're working. So that's definitely a topic we can all relate to. I'll start it off with the first question, and if anyone else has a question after that, please follow suit. But if you're an organization that doesn't do this, because this is quite specific and quite novel, how can someone get started improving their reef grazer population? Is there something that can be done that's a really easy starter step?
Yeah, that's a question that many people ask. I think the simplest things to start is looking what is there, because before we are starting enhancing, we have to know like if this is actually needed. So I would say that is the first step, and that of course only requires like some fieldwork equipment, so it's relatively easy. And then next steps all require some kind of head starting lab equipment to run a couple tanks. It can be relatively easy. We didn't have a fancy lab like we have now. When we started, we had an old 20-foot sea container, and that worked as well. But you need something to let them grow.
Great. Does anyone else have any questions out there?
I have a question. Hello, Alwin. Thanks for your nice presentation and very interesting work that you're doing in Sabah and the wider Caribbean, hopefully, if the project works. I was wondering more from a fundamental biology perspective. So if you see the Caribbean as a big food web, and now there's more algae than in the past, so you would say there's more food available for grazers, but not for the diadema, because there are some like these diseases go around. Why do you not see a natural revival of like the populations of these white sea urchins of whelks on their own? Because you could expect if they eat these algae, that by themselves their populations should grow automatically, right? Or so why would you need to introduce them? Why doesn't it go by itself? Also, because if you know why they don't grow in their populations by themselves, that could also be a reason why restoration would also maybe not work in the end. I'm just I'm not maybe a bit chaotic.
No, I understand the question, and it's something that puzzles us also a little bit. I think it's not one factor, but it's probably multiple. But the same is true for why did the diadema population simply did not recover. I mean, these are super fertile animals. Like one female can produce 17 million eggs. Like the expectation after the 1980s was that this population would bounce back within a couple years. And that didn't happen. And one of the things that we're actually exploring now with population modeling is that these populations have sort of multiple stable states, either at a very low, like 0.1 individual per square meter, stable density, or very high, up till 10 individuals per square meter. And to get from the one to the other is pretty hard for a population. Like even at 5, for example, this is all hypothetical, but they could still go back in a couple of years till the 0.1. And only if they go beyond a certain point, the increased density result in a relatively reduced predation, partly because there's simply more individuals to choose from for a predator, but also because they start modifying the habitat in a positive way, and then the populations start increasing again. And it's not a complete answer to your question, but I think most of the invertebrate grazers have all experienced certain sort of bottlenecks in the past. So the diadema had the die-offs. The white sea-urns were very heavily overfished, especially on the French islands. The whelks, the Sepika, they are probably the oldest fish species in the Caribbean, because you can just walk into the water and take them. So even before people started fishing underwater, they already fished on these species. And the herbivorous fish, of course, also have been historically overfished. So there's all kinds of different reasons why things are not coming back naturally. And it is something super interesting and something that I would like to explore further, and that can also help us in restoration, because if we know at a certain moment what this sort of tipping point is, then we know how many we need to be able to keep there. Because it's pretty clear, if you put like 100 out, they will stay around for a while and they fulfill their fantastic ecosystem function. So they enable coral recruitment, but they do not enable new population. Like slowly the numbers decrease again, and then you're back to where you were.
Oh yeah. Okay. Yeah, that's an interesting perspective indeed, and does sound there's enough work to do.
Yeah, absolutely. Yeah.
And it doesn't sound hopeless. That's at least what I also get from it. Like if you know these more fundamental reasons, and then you know maybe around a certain island, which you could see as a micro habit or as a population area for these urchins, if you just dump in sufficiently, then at least around that island it should recover, you could argue.
Yeah.
I guess.
Yeah.
And then maybe you get spillover effects to nearby islands or reefs or anyway.
Yeah, exactly. Yeah.
Thanks for your answer.
Yeah, thanks. And nice to meet again. That's been a
Yes,
while.
it's been a while. Yeah. Good to see you.
Great. Any other questions out there? It is a hopeful vibe from both of these presenters, which is really nice, because I think in conservation we can be a bit negative sometimes. So it's nice to see the hope on the horizon. I see a question from Stephanie. Do you want to ask yourself, or would you like me? I can also ask.
So I can repeat the question.
Hi.
Yeah.
Oh, hey,
Steph.
Stefan.
How are you? Okay, thank you very much for the presentation. So actually my name is Filippo, but I don't know why. I think I logged in with the computer was previously to Stefan, so the name is still Stefan. But anyway, it's just a detail. So I'm from St. Martin and from Nature Foundation, and my question was more related to if there are sea urchin population more abundant in one place of the island. Do you suggest and do you think it's going to be feasible to move them to another place where maybe microalgae are more abundant? So maybe it would be a possibility to fight that problem maybe on the reef of the island or somewhere else?
Yeah, that's a good question. I think you could do that as long as you don't damage the donor population, because if they are established in a certain place, but they don't provide direct ecosystem benefits there, they still spawn, of course. So they provide larvae to downstream areas. So the population might still be pretty important to keep it there. But if you have, for example, 5,000 individuals, then and there's regular recruitment, it might not be a problem to remove 500, for example, of them. So then, yeah, but I would, yeah, with care, because there's definitely a big chance that if you put them in the right spot, very few will survive. So you definitely don't want to damage the donor population, but yeah, it's possible.
Okay, so maybe just first, let's see how many individuals are there right now, and then maybe try to find a proper number of individuals to move to avoid any as you said, damage to population. Because I mean, we're not diving so often the lagoon here in St. Martin because, as you probably know, it's very highly polluted. So, but sometimes where we go there maybe for seagrass monitoring or whatever, I saw last time that many urchins were there present. So I was thinking, based on your presentation, maybe could be a possibility to move few of them out in the ocean, on shore, in the coastline, instead of keeping them all inside the lagoon, where there are also problems outside there. So you answered my question. Thank you.
Yeah, yeah, yeah. Do it with care. Otherwise, you end up. They have their function in the seagrass as well. Otherwise, you end up with two places without sea urchins if you move them to
Yeah, no,
a suitable place.
Definitely not. Okay. Thank you.
Great. I think we have time for maybe one more question. There's one out there. All right. Well, if questions come up later, again, feel free to reach out directly either to us here at TRN or to Alwin. Maybe Alwin can put his email address or the project link in the chat for anyone who's interested. And again, this has been recorded, so we'll upload everything onto YouTube here in the next couple days. So if you want to revisit it or reach out to either Alwin or Sander, that will always be an option for you. So again, I'd like to thank everyone very much, especially to our presenters for coming. And I hope everyone has a great day. Thank you very much, everyone.
Thank
Thanks,
you.
Jessica, and thanks everyone for attending.
Thank you very much. Have a good one.
Thank you.
Bye.
Yeah.
Bye, guys.
Speak soon, Alwin.